Hello and welcome to the Photosynthesis 2030 webinar series, a series aimed at promoting collaboration across the photosynthesis community. It's funded by Capitalise, a Horizon 2020 project that uses natural variation and innovation to improve photosynthesis to create higher yielding plants. I'm Dr. Louise Diva and I'm very pleased today to welcome our two presenters, Professor Andreas Weber and Dr. Stéphanie Arrivault. During the talks, don't forget to put your questions in the question box. And after the presenters finish, I will put the questions to them. Our second speaker today is Dr. Stéphanie Arrivault from the Max Planck Institute for Molecular Plant Physiology in Potsdam. And her work focuses on quantification of metabolites in the Calvin-Benson cycle to study regulation and interspecies diversity. She also does 13 CO2 labelling to study fluxes in C3 and C4 photosynthesis and is involved in the C4 rice project. Today, she's going to talk to us about targeted metabolite profiling as a top-down approach to uncover interspecies diversity in the Calvin-Benson cycle. Okay, thank you.
Thanks a lot, Louisa. Can you see my screen?
Yes.
Oh, perfect. So it turned out that the two presentations are quite fitting together. So a few things that Andreas mentioned, I will also mention, but then extremely shortly. So first of all, thanks a lot for inviting me to give this presentation. I will talk mostly about the Calvin-Benson cycle and measurement of metabolites in different species. So the Calvin-Benson cycle evolved more than 2 billion years ago. Despite its age, the structure is highly conserved. It's been mostly unchanged from cyanobacteria to angiosperm. And you can see in this representation that the Calvin-Benson cycle has three phases: the carboxylation with incorporation of carbon and formation of 3-phosphoglycerate, then the reduction phase and regeneration to produce ribulose bisphosphate again, which will be used for the fixation by Rubisco. So despite the structure being the same in a lot of species, there are a lot of evidences that there's variation in between species, how it operates. So there's evidences for differences between cyanobacteria and eukaryotes, so mostly about the Rubisco specificity, the structure, compartmentation. Between photosynthetic organisms that do and don't have a CO2 concentration mechanism, also called CCM. So Andreas mentioned that, like organisms doing C4 photosynthesis, also organisms having pyrenoids and carboxysomes, but also in plants performing C3 photosynthesis, so in higher plants. We have also differences in photosynthetic rate and light responses. Most of the studies so far are focused on single traits, so mostly a lot of work done on Rubisco, such as the kinetic regulation, and concerning plants having different type of photosynthesis, it's about leaf anatomy and ultrastructure. So we wanted to know if we could use metabolite profiling to search for changes in how the CBC operates. So when we have metabolite amount, we can see if they change between the species, and it could highlight changes in the balance between different enzymatic steps. For example, an enzyme at which the flux is simulated can be identified because the substrate will fall and the product will rise. But an enzyme at which the flux is inhibited will be identified because the substrate will rise and the product will fall. The idea is that it will give us insight into the adaptation and evolution of the CBC and give strategies to improve photosynthesis in a given crop. So this part goes a bit with Andreas' part, but not completely, as he mentioned that their strategy is not to use what is already known about Calvin-Benson cycle, but to implement new enzymes. Our approach is still to have more knowledge about Calvin-Benson cycle and during the evolution from C3 to C4. So there are two different approaches, but they fit also together. So quantifying CBC metabolites is not a new thing. It's been done in the 70s and the 80s by a lot of groups over the world. The problem was that only a few metabolites could be measured, and mostly it was due to technical reasons, and it was done by enzymatic assays. Also, mostly only one species was used per study. So what we want is to be able to measure reliably and quantitatively all or almost all the metabolites in the Calvin-Benson cycle. So in the, I will say, the last decades or a bit more, there's been a lot more use of mass spectrometry, and that's what we are, sorry, what we are using to quantify metabolites. So we use LC-MS. We can start from extremely low amount of material. We measure about 30 metabolites, and we have other metabolites measured by additional platform and also by enzymatic assays. We have an extremely good coverage of the Calvin-Benson cycle. We are missing mostly its erythrose phosphate that we cannot measure because the standard is not of really good quality. But when you do measurement with mass spectrometry, there's quite an inherent problem. It's called the matrix effect. The other compounds in your extract will affect the signal of the compounds you want to measure. It can enhance or decrease the signal, so you will have a false read of the amount. In order to correct that, you can use internal standards, so that's what we are doing. We could buy some, or we also synthesize some using isotopically labeled standards. And for example, we cover the hexophosphate, the pentose phosphate, we have also DAP and fructose bisphosphate. Altogether, about 70% of the metabolites we are quantifying have an internal standard. So we have quite a good method, really robust with correction for matrix effect. But there's another point to take into consideration, is the harvesting procedure. When we look at the half time of metabolites from the Calvin-Benson cycle, it's less than 1 second, so it's extremely fast. In the past, we did an experiment harvesting Arabidopsis thaliana to see the effect of how we can harvest. So the plants were grown into a percival. We quenched the rosette by pouring liquid nitrogen on top of it, or we cut the rosette and transfer into liquid nitrogen while everything was done in the percival, so under the same light field, or we took out the plant from the percival and harvested in the corridor. It took only a few seconds. And you can see that some metabolites are extremely affected by that. For example, here, fructose bisphosphate, DAP, the amounts go down drastically when you have only a bit of reducing the light intensity, a bit of shading. So there will be changes in metabolite content if you don't quench very quickly and without changing the condition. Mostly do not change the light intensity, do not even put your hand on top of the plant before you harvest it. In the past, we did already a lot of measurement with various species due to various projects we had, and then we wanted to check if we could compare the species just using the Calvin-Benson cycle intermediate. So we had 4 C4 species, all of them NADP, ME subtypes, and we had 5 species, C3 species. They were grown in non-stressful conditions with moderate light and moderate temperature, and the plants were harvested the correct way. Here you can see metabolite profile of these 9 species. I will not go into detail, but just to see that the profiles are quite different between the species. Highlighted in green are the C4 species. But we found things that we expected. For example, in C4 species, you have a lot less ribulose bisphosphate than a C3 species, because you have this CO2 concentration mechanism. So we did some PCAs to have better visualization of what's happening. In green, you have the C4 species. In black, you have the C3 species, and they separate quite nicely from each other. However, the C3 species in black are a bit all over the place. And we were wondering if it could be maybe an artifact, because the plants were not all growing under the same light intensity. Some had higher light than others. Could it be also due to leaf composition? So we tested this hypothesis. For maize and Arabidopsis, we checked the metabolite profile under different light intensities. And we could see that in general, there's a siren outside. There's good correlation between the different light intensities, so that does not affect really the profile. And when we look at our PCA, we had all the Arabidopsis thaliana plants at different light intensities grouping together, and the same for the maize samples. So then we were thinking maybe it could be due to leaf composition. We noticed that cassava and rice have extremely high level of protein compared to the other species. So instead of having just our PCA done with data normalized on fresh weight, we did also on protein and also on chlorophyll. You can see here the three PCA plots, and we can still see the separation on all of them between the C4 and the C3 species. So it's quite robust. But we were thinking that it's not an easy task to compare really different species. And as we saw with the difference in protein amounts, so we decided to go for another kind of normalization that we call dimensionless. So we calculate the amount of carbon in a specific metabolite, and we take it as percent of the total amount of carbon in all the Calvin-Benson metabolites that we measure. And you can see here this dimensionless PCA, where we have again a really nice separation. And for all future analysis, it's also what we're going to keep. We use the so-called dimensionless dataset. So from this first try, we could really see nice separation of C4 and C3, as I mentioned. The C4 species also separated from each other, and we think it's based on their biochemistry, mostly the presence of a 3-PGA triose phosphate shuttle. So maize is known to have that. Setaria, there's quite big evidence. It also has this 3-PGA triose phosphate shuttle. However, Flavoria, it could be a partial shuttle, so it's not a surprise it separates from Setaria and from maize. The C3 species, even when we use a dimensionless dataset, they still separate at a quite high degree. And we have to remember that any evolutionary adaptation of the Calvin-Benson cycle in C3 species will have occurred independently in each plant lineage, and will probably have been influenced by specific factors relating to prior history and environmental nature. What it also shows us that the best strategy to improve C3 photosynthesis might vary from crop to crop when working with the C3 plants. They do behave differently regarding the Calvin-Benson cycle. So we wanted to add more species to our analysis, species having also the CCM. So we used the Flavoria genus, where you have intermediate from C3 to C4. And also we use some alga and cyanobacterium having cyanobacteria having pyrenoids and carboxyls. Starting with Flavoria, so as I mentioned, you have a spectra for the evolution from C3 to C4 photosynthesis in this genus. You have the C3, C4 intermediates, which are sometimes called C2 photosynthesis. You have also C4-like. When we look at the tree of the Flavoria species, you can see that there's a clade A with a lot of species and a smaller clade called clade B, and the photosynthetic type are distributed between these two clades. With a little asterisk, I indicate the plants we use in this study. So we cover some C4 that we did already have in our previous study, some C3, C4, and some C4-like. And also we have some C3 species from Flavoria. So to our previous analysis, we have now 2 additional C3. We have 2 C3 and C4 intermediate species, 3 C4-like species, and we have, as I mentioned, we already had the 2 C4 Flavoria species. And here is a PCA plot showing the photosynthetic type mode with different colors. But what we can really see that the C3 Flavoria in black, they are grouping with the other C3 species. They kind of lie in the middle of them. The C3, C4 intermediate species in light blue, they diverge from that, and also the C4-like, and they are both in a trajectory going to C4 photosynthesis. And we can really see that CBC adjusted progressively during the stepwise evolution of C4 photosynthesis. You can also notice some of the samples from clade B, so also C4-like. They are also in this trajectory going direction C4 photosynthesis. However, they are separated from the other due to the evolution on this tree. So we can, even with the PCA, see the different part of the tree is a different clade. And if we look at what is driving the change from C3 to C4 is to have less 3BP, also less FBP, SBP, F6P, and S7P, but also, we also see more 3-PGA and more triose phosphate. The other species we included are alga and cyanobacteria. The idea behind that is they also have this CCM, and in the literature there's not that much information about metabolite amounts. A lot of work has been done long ago by Bassams and colleagues. Lately, Tabea Metler and Anna Kuken. There's not that much. So we are thinking we can also increase the knowledge about Calvin-Benson cycle in such organisms. It's a collaboration with him Tribes, who was at the Max Planck Institute before, but now is in Tel Aviv University as a senior lecturer, and also with Martin Egemann and Stefan Lucius from the University of Rostock. So we have 2 Chlorella species and Chlamydomonas, and we also have Synecocystis. They were grown in ambient CO2 and in low light, so they were in CCM-inducing conditions. If we look at the metabolite profile, so I put back again the C4 species, the C3 species. In pinkish are the alga, and something extremely striking is an extremely high level of 3-PGA in this alga. It's extremely high. We even had to measure with different methods just to check that we were not wrong. If you take out the 3-PGA from alga, the highest metabolite in the species is ribulose bisphosphate. And when we look at cyanobacteria, the highest metabolite in there is also ribulose bisphosphate. By doing a PCA, we could see that the species are really separating from the C4 species in green, and they also separate from each other. There's been this study I mentioned from Tabea Metler and Anissa Kuken. They predicted by doing some modeling that the substrate of Rubisco 3-PGA, the substrate of Rubisco ribulose bisphosphate and the product 3-PGA will have really high fluxes in CCM-induced cells. And in fact, it's what we are seeing, that these 2 metabolites, ribulose bisphosphate and 3-PGA, are really driving this species away. So even if they do perform CCM, or they have a CCM, it acts completely differently than a plant. So altogether, we could see that we could use this methodology to see interspecies variation in how the CBC operates. We are using a dimensionless dataset, so it helps us to have non-biased comparison between the species. We did more work with this data. For sake of time, I didn't really want to show that, but we did also covariance analysis. We also do a metabolite ratio, is to show exactly which enzymatic steps show interspecies variation. So I would direct you to some of the paper we published lately. We are in the process of expanding the species we are working on. It's a PhD project from Vittoria Clapero in our group. So it will be some additional C4 species, as so far we have only the NADP-ME subtype. And we will use also Neuracne in a similar way as we use Flavoria, because this genus also have different photosynthetic type, the C3, as you C4-like, and the C4. And we will continue to do some work with alga and cyanobacteria, this time in non-CCM inducing conditions. To finish, I would like to thank the funding grants helping us to do this work. Also Mark and John, the people who are involved in doing experiments, doing some measurements also. And thank you for your attention.
That was great, thank you. The questions are already coming in. Did you try to visualize the data, including the 3rd principal component? Perhaps this can give a clearer separation between samples. That's from Ivan Renaya Iliorans. I know that's a bad pronunciation, whoever that was. So did you try and visualize the PC3?
Oh, yeah, we did that. We did different variation of the PCS we were taking. Just for the sake of the talk, I kept the PC1 and PC2. But yeah, we tried also other variations. And generally, we see always the separation with the vectors really showing the same. And it's true, in some cases it's more obvious than in other cases when we change the PCAs we are taking into consideration. Yes. It's just I wanted to keep it kind of simple.
Yeah. Okay. One from me. Do you think you talk about different crops having sort of different metabolites in the CBC cycle, but do you think there can ever be an optimal CBC cycle? That if you were a plant breeder, you see, as a plant breeder, it's quite damning, isn't it, to say actually you can optimize, say, SPPAs, whatever, but if it's a different species, it might not work. So do you think there could be an optimal CBC cycle that would work for at least C3 plants or C4 plants?
I would say it's a tricky question, because also it will also depend on which condition you're going to put the plants and how that will react to the conditions. So it will be really hard to have a common CBC for a lot of plants, sadly.
Oh, hold on, I'm just having to change my screen a second. Okay. And would you say that, I don't know, this is a really naive question I'm asking you, would you say that sort of metabolome assisted breeding was something that would be a forward-thinking way to, to go? It'd be obviously extremely expensive, I presume, but so you're phenotyping, genotyping, and metabolome. Would that be the way forward potentially in terms of looking at you?
That will be tricky to do. I think it's really nice to have knowledge about the species you are working on and the one you want to improve. And if you manage to improve some of them by transformation, it would be really nice also to check the metabolome in this transgenics. But it will have to be kind of really, you need to specify on which plants you are looking at, because you can't do that at a really, how you call that, you can do extremely big screening. It's extremely time consuming, but yeah, it could be done in parallel with lines of interest, but not for screening.
No, I suppose that would be an awful lot of work. Is there any other questions anyone would like to ask? Please type them in the box now if you would like to. Have you thought about comparing your plants because you grew them under non-stress conditions, didn't you, when you compared them, so they were optimal? Do you think if you've grown them under stressed conditions, you might have them clustering a little more? For example, they'd have a similar response. So is that a possibility, or is that nonsense?
I mean, what we did with maize and Arabidopsis, the different conditions were not stressful enough for them, but it's a possibility. It might really bring more changes. Plants will probably not be extremely happy. We are doing some extra work on maize with different light intensity and just checking the changes. In general, as we want information just about how the Calvin-Benson cycle works in a happy plant, we stay with moderate light and moderate temperature.
Okay. Well, if there is no more questions, this is your last chance if there is questions, but if there is no more questions, yes, then I would like I'd like to thank our speakers. Thank you, Stephanie and Andreas. Oh, hold on. There's a question. I'm afraid I have to stop. There's a question. This is from Christine Rains. What do you think is causing the differences, regulation or enzyme kinetics?
I think it's everything related to the redox, but With our data, it's really difficult to answer this question. We can just check which enzymes will work more or will work less. To say what exactly is doing, probably redox.
Okay. Well, thank you. And I'm gonna thank you both for very stimulating talks. And to draw your attention to our next month's webinars, presentations from Marjorie Lunsden and Virginie Mengin on the 5th of April next month. So hopefully we'll see you there. Don't forget to register. So thank you very much once again to our speakers. See you all again soon. Bye-bye.